Biology, paper 1. Question 1. What is the difference between aerobic respiration and anaerobic respiration in yeast? The answer is C. Anaerobic respiration produces ethanol, aerobic respiration does not. Question 2. If a plant is exposed to light, which color of light would lead to the lowest rate of oxygen release in green plant? The answer is C. Green. Question 3. A 2-cell sea urchin embryo was physically separated by scientists into 2 cells, blah, blah, blah. The answer for that would be, the question is, what is the relationship between the 2 adult sea urchins? The answer is D. Both adults would have haploid cells. No, cross that out. They are clones. It's D. 4. Which genotype would be normally found in gamete? The answer is B. Capital R and S. Which statement applies to meiosis and mitosis? This is question 5. The answer is B. Separation of chromatids occurs in both meiosis and mitosis. This is question 6. The hydrolysis of pure sample of an organic molecule produces a pentose sugar. Blah, blah, blah. The answer is C. Phosphate. 7. The graph shows the results of experimental investigation. What can be conducted from these results? The answer will be D. The percentage of sugar remaining after 42 hours is greater for galactose than glucose. Question 8. What features of eukaryotic chromosomes distinguish them from the chromosomes of prokaryotes? You're gonna write A. Circle A. Histone proteins. Question 9. What benefit is derived from the use of Bt crops? You're gonna write D. It can lead to a reduction in the use of pesticides. 10. Which feature is common to both mRNA and DNA? You're gonna write A. Answer is A. Covalent bonds between adjacent and nucleotides. 11. What is the reason for tag DNA polymerase being used in the polymerase chain reaction? The answer is A. It does not denature at high temperatures. Question 12. Plants produce carbon dioxide in respiration and use carbon dioxide in photosynthesis. You're gonna circle D. 12 is D. There is more respiration than photosynthesis between W and X. 13 is the pedigree chart. It's A. Inherited as dominant autosomal allele. 14 is D. All dwarf plants were homozygous. 15 is C. A, B, A, and B. So it's like circle the C. 16, circle B. 17 is D. 18, C. C is lactate. 19 also C. 19 is like C, right? Up and absorb CO2. And then 20, what does this show? No. Yeah, 20, what does this show? You're gonna select D. The child is male without Down syndrome. Paper 2, 1A. Photosynthesis is process Y. Process Y, right? Photosynthesis. Photosynthesis. Organelle is chloroplast. CHLOROPLAST. Part B, I said name the Z one. You're gonna write glycerol, GLYCEROL. C, part C. Condensation. D, aerobic respiration. Aerobic respiration. E, you're gonna write ATP in capital letters. ATP. Can be used as an energy source for future references. Comma. It releases energy for biochemical reactions. Comma. It also has active transport. Okay, so I'm going to repeat again. ATP can be used as an energy source for future references. It releases energy for biochemical reactions. It also has active transport. Part F. There is a breakdown of monosaccharides. MONOSACCHARIDES. Okay, now 2A. The diagram shows the stages in the cell cycle. You're gonna write, for part A, there are a few processes that occur during interphase. Such as the replication of DNA. Comma, the growth of the cell. Comma, protein synthesis, etc. For part, okay, I'm going to repeat it again. There are a few processes that occur during interphase, such as the replication of DNA, the growth of the cell, protein synthesis, etc. For part B, you're going to put the letter C on prophase in the graph. Prophase. For part C, you're going to put M on anaphase. Okay, now in D, you're going to write, in mitosis, there is a production of two daughter cells whereas in meiosis, meiosis, there is 4. So in mitosis, there is a production of 2 daughter cells, whereas in meiosis, there is 4. Mitosis produces genetically identical cells, whereas meiosis doesn't. Mitosis produces genetically identical cells, whereas meiosis doesn't. Comma. Or full stop. Mitosis also produces cells with the same number of chromosomes bracket 2n, bracket closed. But in meiosis, they are in half. In the bracket, write n. Okay, so in mitosis, there is a production of 2 daughter cells, whereas in meiosis, there is 4. Mitosis produces genetically identical cells, whereas meiosis doesn't. Mitosis also produces cells with the same number of chromosomes, 2n, but in meiosis, they are in half, n. Okay, for E, you're going to write mitotic index equals. Then draw the fraction line. On top, write number of cells in mitosis. And under it, write total number of cells. Times 100. So mitotic index equals number of cells in mitosis divided by total number of cells. We'll also use. Okay. Now 3a. For 3a, what you're going to draw is the exact opposite of what there is on this side. So the complementary strand. You see okay, so what you're going to do, you're going to make you're not going to draw, okay, next A, you're going to write T in a box. And then a straight line towards the right side. Then you look at the hexagon connected on the left side. You're going to draw the exact opposite of it. So the pointy area will be facing downwards. And the circle part is going to be connecting to the most right area. The most right area, you have that point that's a little bit downwards. Okay, so A, connect A to T, C to G, and T to A. Do this for all of them. I'm gonna give you like a few minutes for that. Draw it for all of them and connect it. Also, when you're connecting, it's gonna be, you know how the bottom part is on the top, right? Like it's flat there. You're gonna connect it to the left side of, the right side of it. Okay, you do that. 3b. Define mutation. A mutation is a change that occurs in our DNA sequence. A mutation is a change that occurs in our DNA sequence, either due to mistakes when the DNA is copied, or the result of environmental factors. Such as UV light and cigarette smoke. It passes off to offspring. C. As mutations occur, natural selection decides which mutations will live on and which ones will die out. As mutations occur, natural selection decides which mutations will live on and which ones will die out. If the mutation is harmful, the mutated organism has a much decreased chance of surviving and reproducing. The mutated organism has a much decreased chance of surviving and reproducing. If the mutation is beneficial, the mutated organism survives to reproduce. The mutated organism survives to reproduce. And the mutation gets passed on to its offspring. Full stop. In this way, natural selection guides the evolutionary process to incorporate only the good mutations into the species. And expunge the bad mutations. I'm going to read this C part again. As mutations occur, natural selection decides which mutations will live on and which ones will die out. If the mutation is harmful, the mutated organism has a much decreased chance of surviving and reproducing. If the mutation is beneficial, the mutated organism survives to reproduce, and the mutation gets passed on to its offspring. In this way, natural selection guides the evolutionary process to incorporate only the good mutations into the species and expunge the bad mutations. Okay, now for A, we're going to draw a molecular demand of alpha-D glucose. Okay, I'm going to tell you what to draw. We're going to draw like the hexagon thing, but it has 6 points. So draw it. So you draw like opposite facing arrowheads like horizontally from each other, and then you draw a line that connects the side thingies. Okay, once you did that, on the top, we're going to start from the top, what we're going to write, okay? So the top, the furthest to the left one, you're going to draw a vertical line a little bit up. And you're going to write C, H, and under H, write a 2, and then go back to the normal writing thing and write OH. Okay, next one. We're going clockwise. The next point on the top, you're going to write O, like an O there. Next point, this is like the pointy side to the right. You're going to draw a vertical line down and label it as OH. Do the same to the one next to it as well. We still keep going clockwise, so the right side of the bottom one. OH, and then the one on the left side of the bottom part, you're going to draw a vertical line up. It's a little bit in the 6-side hexagon thingy, and you're also going to write OH for that. And then for the point of the left one, you're going to draw a vertical line down and write OH. Okay, now for B, right? This happens by the process of photosynthesis. This happens by the process of photosynthesis. Comma. It occurs in the chloroplast. It occurs in the chloroplast, CHLOROPLASTS, chloroplast of plant cells using chlorophyll. Of plant cells using chlorophyll, CHLOROPHYLL, comma. Chlorophyll absorbs red, blue, and reflects green light. Chlorophyll absorbs red, blue, and reflects green light. Then you're going to write, raw materials are carbon dioxide and water. Raw materials are carbon dioxide and water. Oh, you scared me.
Saw that.
What?
Kim tänker
Sana. We're just talking. Eşit mi? Tamam da biyoloji danışıram.
Yemeği hazırdı.
Geldim. Okay. Raw materials are carbon dioxide and water, comma. Water is split by photolysis. L-Y-S-I-S. Water is split by photolysis, comma. Oxygen is produced as waste. Oxygen is produced as waste, comma. Glucose is then formed. So oxygen is produced as waste. Glucose is then formed. And then another comma. Glucose molecules combine to form. Glucose molecules combine to form. Starch for storage. Starch for storage, comma. Light energy is then transformed to chemical. Light energy is then transformed to chemical. Light energy is then transformed to chemical. Question 5. So 5A. You need to draw a ultrastructure of a prokaryotic cell based on electron micrographs. Okay. First, we're gonna draw a big, square. Make the edges a little bit curvy. A large, curvy square. Make it a little long. Okay, you're gonna draw another one inside of it. Make sure it's kind of close to the edges, but not too close. And then you're gonna do another one, but this time do it really close. Just to the edges of the inside and everything. Okay, so now what you're gonna do from the top, from the left side, the corner one, you're gonna do another one. And when you reach the top, what you're gonna do is bring it a little bit midway, right? And then you're gonna draw kind of like an opposite Y shape of it. It's kind of like 3D thingy. It looks like two ball sacks hanging, okay? I'm gonna draw that. And then next to it, on the left part of it, you're gonna draw like 6 dots. And then label the dots as 70S ribosomes. Okay. And then below the freckle type of dot thingies, you're gonna just draw random shapes of long stuff that are going in a weird McDonald's M type of thing, and then the opposite of that as well. Make it in 3D. And then you're gonna label that as mesomome. M-E-S-E-M-O-M-E. Mesomome. Okay, go down. And below that, you're gonna draw 2 circles. First one should be big, the little one to the little down left side should be a little smaller. And then you're gonna label the big one as plasmid. Okay, now we're at the complete down part of the cell thingy. What we're gonna do from the cell line, the line of the most inside of the cell, we're gonna bring that down and then kind of like draw like a tail shape with it. And then you can just erase it, like the inside part of it, because there's lines there. And then you're gonna label it as flagellum. 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 Okay. And then, from the opposite side to the symmetrical side of the weird shape that we drew, like the M McDonald's thingy, you're gonna draw a double helix one. And then you're gonna label that as DNA. Also, next to the flagellum, you're just gonna like draw a kind of like a 3D line thingy that goes inwards to it, so it looks like a hard penis standing out inside it, and just delete the little line that's left in the middle thing. Okay, after you've done that, outside on the outer square one, you're gonna draw just like little floaty S's, like attached to it, little floaty S's. Label that as pili, P-I-L-I. Okay. Okay. Now we're gonna label the rest part. All the inside area that's left free inside it, you're gonna draw an arrow to it, and then label that as nucleoid. 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 And then, okay. Let's start from the first square, like the outer square, connecting with the second outer square. The middle part of that is called capsule. Capsule. The one after that is called cell wall. And after that, it's called cell membrane. Cell membrane. Yeah, and that's what you do for 5A, and then 5B. Outline what occurs in cells during first division of meiosis. Number 1. Halves the chromosome number. Halves, Halves, the chromosome number. Halves the chromosome number, and then colon, produces haploid cells. Produces haploid cells. Produces haploid cells. Okay, number 2. Beginning of meiosis. Beginning of meiosis, comma, each chromosome consists of 2 sister chromatids. Consists of 2 sister chromatids. attached by a centromere. Attached by a centromere. CENTROMERE. Number 3, prophase 1. Colon. Pairing of homologous chromosomes. Prophase 1 is pairing of homologous chromosomes. Number 4, crossing over occurs. Number 4, crossing over occurs. Number 5, chromosomes condense by supercoiling. Chromosomes condense by supercoiling. 6, metaphase 1. Colon. Pairs of homologous chromosomes slash bivalence move to the equator of the spindle. Okay, so number 6 was metaphase 1. Pairs of homologous chromosomes slash bivalence move to the equator of the spindle. Number 7, anaphase 1. Spindle fibers pull chromosome. Spindle fibers pulls chromosome to opposite poles. Spindle of fibers pulls chromosome to opposite poles. 8. Telophase 1. Telophase 1. Colon. Arrival of centromere with sister chromatids. Arrival of centromere with sister chromatids at opposite poles. Arrival of centromere with sister chromatids at opposite poles. Okay, now 6a. You're going to write man with Down syndrome. Man with Down syndrome, and for B, you're going to write 21 and then Down syndrome. 21, Down syndrome. Seven, you're going to write, firstly, DNA strands. Firstly, DNA strands are separated by the RNA polymerase. Firstly, DNA strands are separated by the RNA polymerase, comma, then new nucleotides get attached. Then new nucleotides get attached to the template strand by RNA polymerase. Firstly, DNA strands are separated by the RNA polymerase, then new nucleotides get attached to the template strand by RNA polymerase, comma. Next is complementary base pairing. Next is complementary base pairing, thymine with adenine. Thymine and adenine, and then adenine and uracil, and then cytosine and guanine. Okay, so thymine and adenine and uracil, cytosine and guanine. Full stop. Then the mRNA will get detached from the template strand. Then the mRNA will get detached from the template strand. And finally, DNA will rewind. Then the mRNA will get detached from the template strand, and finally DNA will rewind.